Hi everyone, and welcome to today's webcast. Our focus today is on LC-MS analytics with a particular spotlight on the oligonucleotide class of molecules. My name is Oliver Ockle, and I am a product and service manager at Vetter. Before we dive into the main topic, I will give you a brief introduction and an overview of our product and service portfolio at Vetter. Please feel free to ask questions in the chat widget throughout the webcast. Our experts will address them afterwards, time permitting. I'm very pleased to introduce our two experts for today's session. First, Melanie Cerula-Wernitz. She heads the Analytical Science Laboratory at Vetter and focuses on analytical development for both clinical and commercial drug products. Second, Thomas Schädle. As head of quality control, his expertise lies in the control and testing of filled and finished units at Vetter. As both of our speakers work in analytical functions that address the day-to-day questions and challenges of a drug product CDMO, they form an ideal expert team to explore our today's topic in depth: how LC-MS can support the oligonucleotide lifecycle. And now some very short information on Vetter. Vetter is a globally active CDMO with both clinical and commercial manufacturing sites worldwide. Our fill and finish sites are closely connected, and each location is equipped with analytical laboratories operating under a harmonized quality system and supported by broad scientific expertise. This structure supports that comprehensive analytical services under GMP and non-GMP are available to our customers for drug products filled at Vetter at each site. Vetter offers a wide range of product-specific and compendial analytical methods, as well as stability studies covering the entire product lifecycle, from drug product development through aseptic, clinical and commercial filling to assembly and packaging. More than 80% of the molecules filled at Vetter are biologics. In recent years, we have seen a strong increase in demand for specialized analytical methods, such as LC-MS analysis, starting already in the early development phase. And with that, I would now like to hand over to our expert on development, Melanie.
Now I'm unmuted. Good afternoon. As said, my name is Melanie Cerula-Wernitz. I'm the head of the Analytical Science Laboratory, which is a development service laboratory at Vetter Pharma. And I would like to start with the global trends on therapeutical oligonucleotides. So in the very beginning, in 1989, the first oligonucleotide was approved. And then there was a small stepwise approach. So 2004 was the second approval, and it went on and on. But nowadays, more than 200 clinical trials are ongoing for therapeutical oligonucleotides. The drivers are indeed infectious diseases in general, but everybody knows the COVID-19 pandemic was the biggest driver of that. And for sure, you can see that the therapeutic oligonucleotides are a technical or technological innovation. In this picture, I want to show that the therapeutic oligonucleotides are used in a wide range of areas. So this can be from metabolic disorders to oncology, but also to dermatology, immunology. And I don't want to picture especially every disorder or every area, but there is also a review article which is referenced here, and this is open access, and this is a very nice review article where you can get a very good overview on what kind of oligonucleotides and what kind of areas are touched here. So now let's make a small trip into biology. What are the oligonucleotides and what is important if you want to analyze them? So first of all, the molecular structure of oligonucleotides. Oligonucleotides consist of so-called nucleotides. These are molecules consisting of a phosphate, a sugar, and a base. And they can differ if you build a DNA or an RNA, it's a desoxyribonucleotide or a ribonucleotide. And if you check the picture here, the sugar differs between these two molecules. And there are 5 different bases: the adenine, the guanine, the cytosine, and thymine. And for RNA, uracil is used. I'm sorry. And if you now build an oligonucleotide out of these nucleotides, it's like putting pearls on a string. So you have a so-called backbone. This consists of the phosphate, the sugar, the phosphate, the sugar. And then you can build a kind of a ladder with the phosphate sugars on the left and the right-hand side, and then the bases in the middle. And it seems like a little bit like a ladder. And this is then a double-stranded oligonucleotide or a nucleic acid. If we talk about oligonucleotides, we talk normally about nucleotides with approximately 20 a length of 20 parts. It could be a little bit less, it could be a little bit more, but it's nothing with 1,280 nucleotides. It's small and short. And you can see here that the double strands are by non-covalent binding paired via the Watson-Crick base pairing. And on the next page, you will see immediately that if you have this ladder and you curl this ladder, this gives the very famous, I would say, helix structure. So a double-stranded oligonucleotide is normally formed as a helix. A single-stranded is half the ladder. And you can also see here very specific aptamers. These are single-stranded oligonucleotides, which builds up a very specific 3-dimensional structure to target any kind of molecules, for example, proteins. What are the challenges by working with oligonucleotides? So oligonucleotides are in their natural environment very unstable. So meaning, for example, on the skin or in the body. So if we talk about the therapeutic oligonucleotides, when you want to administer them, they are unstable in the cells or in the body or in the patient, because our cells learned over the last 100,000 of years that a foreign DNA or RNA is normally something which harms our cells. It's normally a virus. And therefore, there are RNases or DNases everywhere, which makes the oligonucleotides unstable. Not in an aseptic environment. So this is not a challenge for manufacturing. This is really a challenge to get the therapeutical to get the therapeutic dose into the patient. And additionally, oligos do have a very weak intrinsic binding affinity. They are not tissue specific, and they have a very, very low penetration capacity into cells. So this means in the body of the patient, an oligonucleotide is somehow blind. It can't find the way to the tissue where it's demanded. So this means that for therapeutical oligonucleotides, there are a wide range of multiple chemical modifications that the oligonucleotide finds, for example, the way to the liver and can penetrate into the cells. And this makes the analytics sometimes even more challenging. So now, how do the oligonucleotides work in the body? So first of all, there is the standard dogma of the biology. You have the DNA, this is transcribed to the mRNA, this is translated to the protein. If you have a correct DNA, a correct mRNA, a correct translation into the perfect protein, then everything is fine. If you have too much of a protein, a low concentration of a protein, a not correct transcription or translation, this could end up in any kind of disease. And the therapeutical oligonucleotides inhibit specific pathways in this cascade. for example, the decoy oligodesoxynucleotides, these are short double-stranded DNA molecules. They can bind to the genes which should be transcribed, and they can mimic transcription factors so that the transcription is inhibited. Or you can also inhibit the second step, the translation. This could be done, for example, with ASOs, short single-stranded DNA molecules. It's called antisense oligonucleotides, which can bind then to the already existing messenger RNA and leading to the degradation of this RNA. Or you can, for example, use specific SI, small interfering RNAs. These are double-stranded RNA molecules, and this binds also to the existing messenger RNA and leads to cleavage of this RNA. Or, as I already said, the last step could be influenced by aptamers. These are the 3D specific structures of single-stranded DNA or RNA molecules, and they bind very selective and with a very high affinity to all kind of target molecules, normally target proteins, and they prevent then the bioactivity of these molecules. So now this was the deep dive into the biology. Now let's go to the analytical part. What kind of methods, analytical methods, do you need for oligonucleotides? So this differs or by the characterization level or by the production step which needs to be analyzed. So it's a difference if you need to analyze a drug substance or a drug product. And the methods could also differ if you use them for in-process control or for release testing. But in general, you need to verify the following parameters. That's the identity, the purity and or the impurities, the strength, the structure, and for sure the physical chemical properties. Here in this table, you can see where an LCMS can help you with the analytics for oligonucleotides. So for sure, in purity analysis, it can help. If you need to identify impurities, if you need the sequence confirmation, and in the ID testing. So it's a lot of parameters which could be tested or analyzed with the use of LCMS instruments. And if you're now in the situation that you want to buy a new LCMS instrument for your laboratory, there are 4 main different systems of LCMS, and you need to decide which kind of application is the main topic or is the one topic in your lab where you use the LCMS for. So you can differentiate between a single quad or a single quadrupole LCMS, which is a perfect and very robust instrument for identity testing and simple quantifications. Or if your topic is quantification itself, then state of the art is absolutely the triple quad. This is the best instrument if quantification is your daily business. Or if you need more information about identity and no quantification, then, for example, a TOF could be a very good decision. Or if you are in a development laboratory and the question changes from day to day, then there might be a QTOF a good opportunity for you, because then you can get some identity information, you can do structure determination, investigational testing, and quantification. So as said, it differs what your question is, your analytical question in the lab, and what you really need. On the next page, there's more or less a summary of them, what I said already. So as I said, for example, a triple quad is the perfect instrument if you do intensive quantification. If you need more or less everything, then maybe a QTOF would be a good decision. But the price of the instrument also differs with the complexity of the instrument. So the more the instrument can do, the more expensive it is, and also the analytical expertise needed to work with the more complex questions and more complex instrument is also higher. In my lab, we decided to buy a QTOF, and therefore I want to really briefly introduce you into what function a QTOF mass spectrometer has. So there are four, let's say, main modules you use. The first module is the ion source, because you need ions of your analytes to make them detectable. So you only can measure the ions in your sample. Then the next part is the so-called quadrupole. This is used to select the results you want to see. So there you can either do a screening and leaf through every ion which is prepared in the electron spray ionization, but you can also focus on specific masses. If you know what you search for, then you can do a focus step here with the quadrupole. Then the next module is the collision cell. Here you fragment the analytes or the ions which has to be analyzed. So there is a collision gas in the collision cell, and with the collision of the ions, you get fragments of your analyte, and you can then in the detector, which is a time of flight, so TOF means time of flight detector, you can measure the fragments of the ions and determine therefore the mass over the charge. So until now, everything sounds, I would say, very good. Okay, LCMS is a little bit of a higher price, but there are also challenges. Not everything is easy. I summarized some of these challenges here because there are some specific challenges for oligonucleotides in LCMS analysis. And one of the biggest thing is that the phosphates I already showed you in the nucleotides are negatively charged. And due to this negatively charged phosphate groups, you have, for example, a lower ionization efficiency, in your electron spray cell, because there could be multiple charge stages established. And so you have more peaks than one, even if you have only one analyte. And as the oligonucleotides are so highly polar, they could stay solved even in the electrospray. So the ionization is additionally less effective. And what also could happen is this adduct formation, because you normally have alkali metals like sodium or potassium in your sample, and they can bind non-covalently to your negative charged phosphates, and this could broaden the peaks and again gives you a less sensitivity. So there are specific challenges with the oligonucleotides in the LCMS analytics. And this is what I said, you need expertise with these kind of substances. But in the end, you get very precise and very good results. So I showed here one very ugly spectra and a nicer one where we added 8 generic oligonucleotides. So these were 8 20-mers. And in the beginning, we had one wide peak. And at the end, we had the 8 peaks differentiated. So it works, but you need to know how you should get there. And what are the use cases of our QTOF in our development laboratory? With a QTOF or with an LCMS, you can detect and quantify impurities. You can detect and quantify known or unknown impurities, and you can do this in traces. For sure, you can detect, identify, and quantify degradation products, which could happen during manufacturing process or during stability studies, so time or storage temperature related. You can determine the structure and you can do sequencing. You can also test for the chemical modifications, which I mentioned before. So the chemical modifications can confirm the completeness. You can confirm that it's there. You can confirm if there are any changes in the chemical modification, maybe during stability. You can monitor your production processes, for example, the cleaning process or compatibility studies. And in the end, you can support method development, optimization, and validation of other methods. And therefore, I will hand over to my colleague.
Great, thank you very much. So now we heard a lot about the theory behind analytics in LCMS for oligonucleotides. So let's see how we get this working into quality control. So the concept is mainly focused on identity testing in quality control for obvious reasons, right? Because LCMS is by far one of the most suited applications for identity testing in quality control. So let's have a look on where do we need identity testing. So where does it take place in first instance? As we already heard, you might first differentiate between drug substance and drug product testing, because there might be some differences in which stage or what do you really need it for. Also, regarding drug substance testing, there might be reference standard testing and such issues as a time where identity testing might fit your needs. So concerning drug substance, which mostly means characterizing the API, so the active pharmaceutical ingredient of the drug product, there might exist different paths that lead to victory. First off, you might consider the stages of lifecycle management, right? So from early stage development to commercialization of a product. During early stage phases of a product, it's most often sufficient to have the API manufacturer, whoever that might be. It might be your organization itself, it might be a CDMO, or however it is structured, that must perform an ID testing, obviously. For us, from a fill and finish CDMO perspective, during early stage, it is most likely sufficient to have a COA from the API supplier to review as part of incoming goods testing. So with no additional analytical tests required at this stage. However, regarding the product lifecycle management, the more we get into the commercial phase, and later starting with phase 3, we conclude that it is mandatory to have an ID test for the API implemented at every site that handles manufacturing actions on your drug substance or your drug product, respectively. So keep in mind that this may differ if we are talking the site which is releasing the API for further manufacturing. So moving on from the respective COA on the API itself, there might as well be various stages where API testing might find applications. So we have, as already discussed, the raw materials of the incoming API testing, where we need a confirmation of ID. We might have ID testing applications during supplier qualification, or if there is a change in the supplier of the respective API. Of course, for release of the API, there must be some confirmation of ID. And also, I already mentioned it in a short sentence, if you use API for reference standards or anything like that, you surely have to just re-evaluate your standards, and there you need an ID testing as well. But not only the straightforward reasons I've listed here, but also some kind of like investigational purpose, or to prevent even mix-ups regarding on the systems you have to prevent this, there might be an ID testing very, very helpful. So, but that really is kind of deviation specific, but it can be helpful at this stages, which is also part of quality control, right? And of course, ID testing also takes place at the final release control stage. That might be filing specific, but from our perspective, there is a lot of filings that need and final ID confirmation after the drug product is manufactured and released. So now that we've discussed necessity of performing ID tests in theory, let's talk a little bit on how to actually get such systems into your lab. So as LCMS can just be considered some kind of special form of chromatographic analysis, we recommend it is best integrated in the existing chromatographic lab workflows. So after you have the respective components assessed, so you need obviously the LC system, the chromatographic system, you need the components of the DMS, which Melanie already highlighted in great detail. And of course, you need a computer to set up your software that you need for your LCMS technique. So here you can see the little single quad that we used at our Vetera chromatographic lab. So this is really in the neighborhood, close to the chromatographic systems we have online to ensure a seamless integration in our chromatographic lab. So let's have a deeper look at the boundary conditions to get the LCMS running in the lab. So first of all, and most important note, do not underestimate the timelines of an LCMS integration project. It really starts with physical requirements. So you really have to make sure beforehand that the facility holds everything you need. Do not underestimate the space such a configuration eats up quite quickly in your lab. And I'm talking specifically like air systems, supply media, supply gases, and also electricity. Some systems, in case of the application you chose, consume quite a lot of power. And in conclusion, a supplier site preparation check is highly recommended, along, of course, the supplier IQ, OQ, which is nowadays often offered in either traditional paper-based or digital versions or something in between. So this is all part like before you really get the system in your lab online. After that, we surely have the most implementation steps. So we need to make sure that the off-the-shelf system is ready to be used in a controlled environment. So this step really is crucial, and you should be given appropriate timeframes in the overall implementation project. Do not forget that there are also data backup strategies necessary to establish. So there might be a reach out to your IT department as well and find appropriate solutions, and this may also take a little time. However, after those things are set, we need a GMP documentation regarding system validation, incorporating hardware and software together. So now you have your system ready, your system validated and everything. What do you need? Yes, you need the methods to run on the system. And this is usually done during a method transfer. So especially from our perspective, where we do not have methods developed in-house, we use a qualified method transfer to get the methods into our quality control. So there are developed methods from our customers. These need to be validated, and we need to have a confirmed transfer that we can use these methods in a quality control environment. And then you also have to keep in mind that the training of the HPLC analysts to use such systems may take a while. Melanie already briefly discussed it. It is a technique that is very complex and that you really need time to be familiar with it, not only the software, but also to get good results on the methods used. Also, with the training and the methods, they're coming like cleaning and contamination procedures where you can make sure that you do not need a dedicated system for every method you are focusing to run in your lab. So now switching a little bit over from the practical side to the regulatory perspective, and there it is, data management. So the management of your data is really among the key factors. And data management begins with setup and configuration of the application software used for the actual analysis. So most application softwares nowadays offer to some degree like a GMP software package, GMP compliance package, or anything labeled similar. It may be labeled differently from vendor to vendor. It's either most likely GMP module, or sometimes it's even called CFR Part 11 compliance or some similar expression there. Either way, we strongly recommend to have a deep review of the opportunities of software packages, because speaking of our own experience, it still needs configuration to certain extents depending on your specific workflow needs there. Important aspects that I want to highlight regarding these topics are user management, which I would label an absolute key factor for running the system in a QC environment. Of course, obviously, there must be some audit trail functionalities, which is pretty self-explanatory, and also signature management of electronic data in relation to your general data management in a chromatographic lab should be clearly addressed. Walking away from the software configuration alone, there's also the general QC part, which has to be focused on and to be considered and kept in mind, referring to you need SOPs to run the system. Of course, you need different SOPs, maybe even. We decided to have like one SOP that is dedicated to the users, so the analysts themselves, on how to run and manage the system. And we have another SOP that covers more of the software side and the administration side of the system, which is really helpful to keep these things together, but on the respective roles at the same time. So some examples that you also need for like QC environmental forms or anything would be paper-based or digital QC form for the project creation management, for custom calculations if you want to have them run in your system, and also how to validate them, column performances, system checks, and everything that is going on in this part of the QC must be kept in mind to use such a system. Again, speaking from experience and already a little bit teased on the previous slide, I really want to highlight certain aspects of the user management by showing you guys a best practice example of user configuration that's not only for LC-MS systems, but might as well be used as a blueprint for general QC applications. So we use basically the same logic behind the user management for every QC system we run in our labs. And we think that is good to see, because this concept is really authority proven in multiple times. It's usually not enough in the current view of authorities to simply have your user management divided into user and administrator. So we need to dig a little bit deeper here. We recommend to structure mandatory steps in the lab, from sample setup to measurement to review and all that's what's happening there, to be set up by different levels or roles between users that might as well overlap to some extent where necessary. For example, let's have a look at how we configured the role of the analyst and the lab expert. So the analyst basically writes instrument methods, can start measurements, which is the main topic of this role. He might sign data and create reports. But there is a small difference in what a lab expert actually does, because he reviews the methods. He can edit result sets and processing methods, and he also can unlock files. While unlocking files might sound scary in a QC environment, I can assure you it is not, because we have full audit trail functionality, and the lab expert, as you can see here, has not the right to actually start measurements. So this is really the key factor on how to divide different roles in your lab to certain processes that are needed to keep the data in check and to make sure that everything is running smoothly. So while the correct user management sets really the foundation of good laboratory practice, we also need to make sure that the data itself is managed properly. So moving away from the user roles alone, let's have a look on data processing in terms of managing integrity of the data at all times. It starts with instrument methods. Yeah, right, instrument methods. You will need them in your system, and they need approval from different stages. We use a best practice example of a 6I principle with 3 levels, meaning created in the first role, which is done by the analyst or the lab supervisor. Then we have a reviewed instance, and then we have approval instance, which are really the crucial steps to make sure the methods are well under control. The same is true for processing methods. We also recommend to use automatic data processing at every stage during research generation that is possible. It might not be possible everywhere, but when you can implement it, really go for it. So from our perspective as a CDMO, reporting is also a topic not to sleep on. We focus on having controlled and authorized report templates to ensure streamlined and also traceable data reporting to all of our customers in the same manner. And lastly, I want to highlight the necessity of a good segregation of duty concept, SOD in short. This is really the first factor usually to be reviewed by authorities. So we need to recommend to really make sure that the administrators that have software configuration access are located outside the lab environment. So this is the key factor to get your roles and also your administrative issues divided here. So I recommend please use these guidelines as a kind of take-home message on how to best implement complex systems like the LCMS into a QC environment.
Okay, thank you very much, Melanie and Thomas, for these insights into the complexity of LCMS and oligonucleotides. Since different LCMS instruments are best suited for specific analytical questions, we at FETA take a two-branched approach. So we use the QTOF LCMS systems for development and single quad LCMS for quality control, as summarized on this slide again. I hope you got interesting insights, and in case you have any questions, please type them in the chat widget. I see we have already 2 questions received. So the first question is, what other methods for identifying therapeutics oligonucleotides are available in your laboratories? So I think this question can be addressed to Melanie.
Okay, so thanks for that question. We do have, in fact, another identification method, which is also very specific and selective. We use the hyperchromicity. It's a melting point method to melt up the duplexes. It's measured by UV, and you measure a melting curve and determine the melting point. And this is a specific parameter for the identity of oligonucleotides. And this is also available in our QC laboratory as well as in the development service laboratory at FETA.
Thank you, Melanie. The next question is, what do you need from customers to use LCMS at FETA? And I think this question we can address to Thomas.
Yeah, sure. Thank you for that question. As I highlighted in some sentences on one of the previous slides, you see the most important factor is, of course, we need a filling project for your product here at FETA to be able to analyze it. So, but then we really need the methods. We really need to know as much as we can from our QC experts about your product, about the method that must be used to analyze it. The methods might differ in a lot of ways, and it's also very, very important to have it in great detail discussed beforehand, because the equipment might be differ to what we have actually available in the lab. And after that, we need a validated method, because method validation or development is not a service we usually offer at FETA. So we need a validated method to be transferred to our labs in a qualified way. This is pretty standard for us. We do this really a lot in terms of QC implementation, but it really depends on the method and the equipment that needs to be used for your product.
Thank you for your answer, Thomas. So I have no further questions in the chat currently. If any questions came up, please just contact us directly and we can go into touch with you and hopefully can help you with your questions. Then we are at the end of this webcast. So thank you very much, Melanie and Thomas, again for this nice insights. And with that, I say have a nice evening, have a nice day, and hope to see you soon. Thank you.